So these are the steps. First of all, production and isolation of DNA fragment to be cloned. So first of all, we have to isolate the DNA fragment which is to be cloned, which is the DNA of interest. Okay. So DNA of interest should be isolated from any bacteria or any plant of a specific. So this is done by many processes, like first the process is gel electrophoresis, in which the DNA is isolated. That we will discuss later. After that, insertion of isolated gene into suitable vector. So what they do is, this is the plasmid, and this plasmid is a vector. Okay, this is the plasmid, original plasmid, and this plasmid is a vector. Okay, we have added a DNA of interest which is isolated from some bacteria. So this is the DNA. So now what happened is, this is the plasmid. Here we have cut with some endonucleases, and we have inserted this one into it. Okay, this is the DNA of interest which is inserted into this plasmid, which is a vector. Okay, this is a vector, this is DNA of interest. After that, this whole plasmid is inserted into other bacteria, which is E. coli, and there are many strains of E. coli which are very useful in the recombinant DNA technology, and now it will act as a host. This is host inside which the recombinant DNA is inserted, and it also has its own chromosome, and after which this E. coli will start replicating this gene or this DNA of plasmid. Okay, and they will develop many of progenies. So up to this we have discussed, first of all, production and isolation of DNA fragment, insertion of isolated gene, and introduction of recombinant DNA into suitable host. So this is a host and we have introduced. Now the selection procedure of transformed host cell and identification of clone containing the desired gene or DNA fragment. So in all these progenies, all the progeny do not have the recombinant DNA. Many of them will have the recombinant DNA, many of them would not have the recombinant DNA inside them. So some selection procedure is done, like antibiotic. We can see the antibiotic resistance. So this we will discuss later. This is a brief introduction. Or plaque formation. Any of this process is used for selection. So in this selection procedure, many of the progeny will die. Only the progenies containing the recombinant DNA will survive, and then these are allowed to grow further, and after which all the progenies which will be developed from these bacteria, these daughter cells, they will contain the recombinant DNA. Now these all will contain the recombinant DNA, and these all will multiply, and these all will express the introduced DNA of interest or introduced gene of interest inside the host cell. Okay, so this is the whole steps of gene cloning or recombinant DNA technology. So this is a very short form of gene cloning or recombinant DNA technology. This is what we are going to study now in a very elongated way. Okay. Okay, now the tools used for gene cloning. So these are the tools which are used in gene cloning. First of all, restriction endonuclease to cut the DNA at a specific site, and these are called the molecular scissor. So restriction endonuclease cut at particular specific site, okay, and therefore it is called molecular DNA. So the restriction endonuclease develop two type of end, which is a sticky end. This is a sticky end, and this is a blunt end. Okay, so this is uniform and this is complementary. Okay, a suitable DNA molecule capable of self-replication, which is called vector. After that we need a vector, which is plasmid. The most common vector is used is PBR322 and PUC18 and 19. So these are the vector which are very ideal vectors which are used in recombinant DNA technology. And why these are so ideal, this we will discuss in our later videos. Okay, so these are the vector which carry the DNA of interest. Okay, DNA ligase to seal the nick or end that remain in recombinant DNA molecule, it is called molecular glue. So DNA ligase is what actually DNA ligase do is it seals, like this is the DNA of plasmid and this is the DNA of interest. Okay, so now the DNA ligase will come and glue both the DNA or from the other organisms. This will came from some other bacteria, this plasmid is came from some other bacteria, and this DNA of insert will come from some other bacteria. And what DNA ligase do is glue them up. Okay, and that's why it is called molecular glue. Some phosphodiester bonds are there which are glued by the DNA ligase. Okay, then the other one is reverse transcriptase, which convert RNA to DNA, is used to produce complementary DNA copies. Usually mRNA are used for the creation of cDNA library. So what is cDNA library? We will discuss later. So what is reverse transcriptase do? Like this is RNA. Okay, this is single strand. Now this single strand RNA is converted into the double stranded DNA with the help of reverse transcriptase. So this RNA will, like this, these are the RNA, and then what reverse transcriptase will do, they will produce a complementary strand and make it to DNA. So these are the tools used for gene cloning. Okay, so these terminology will be used in our later videos. So these are those terminologies which are regularly used in gene cloning. Okay, now nomenclature. The nomenclature of restriction endonuclease are followed by the general pattern. For example, if we take EcoR1, okay, so this EcoR1, in this E is the genus, the CO is the species, and these three are written in italic. As you can see, these three are written in italic. Okay, and this is Escherichia coli, and this R indicates the strain. Okay, R strain or S strain. So this is the strain, and this I indicate there are three main group of restriction endonuclease, which are type 1, type 2 and type 3. So this is the type 1. So in Roman letter it is written type 1 of endonuclease. So there must be type 2 like in Hind 2, Hind 3. Okay, this is the type 2 endonuclease, this is the type 3 endonuclease. H is the genus name, which is Haemophilus. IN is the species name, which is influenza. D is the strain name, and type 2 and type 3 is the type of restriction endonuclease. So all the restriction enzyme are designated by a symbol R, which is prefix to their name. So prefix to it is R. R equal R1. So what does it mean? That it is a restriction endonuclease of organism E. coli, strain R, and type of endonuclease is 1. Okay. Thank you for watching this video. Subscribe Exam Hai on YouTube. Like our videos and please do comment. Thank you.